Okay, so I want to talk a little bit about your final project. I know you just finished your other project, but it's time to start thinking about it because this is a rapid class. So what I want you to do is really relate how evolutionary theory and the practices, like the actual things that we've been learning how to do, which you'll learn more how to do as we're going, relate to your prospective field. So depending on what you want to do, I would argue that evolution is all around us, right? So there's going to be something that you'll use to inform your decisions in your field. So that is whether or not you're a doctor or a dentist or something like that. You could look at case studies of different organisms and how their evolutionary history better informs your diagnosis. And so we're going to look at an HIV project, and that'll kind of help you understand this case study scenario. There's quite a few of them. We will have talked about Ebola a little bit, but you could even use that one, or various aspects of CRISPR, of these genomic islands, actually, that could be used to use for your study. So what I want you to basically do is find a paper or a case study, like a research article, that really relates to something that you're going to do in the future. Now, if you are, say, going to graduate school, doing undergraduate research, something like that, and you want to pick your study organism, that's great. Do that. We can work with that as well, obviously, and you'll just be talking about the evolutionary history of that study organism. In addition to this paper that you're going to find, which you kind of want to do, like, what is the current state of knowledge of the evolutionary history of whatever organism or system you want to work in. Okay? But then I want you to add to that by, say, finding sequences, aligning them, and comparing them to the existing evolutionary hypothesis. So if you are working with C. diff, you could find similar sequences to C. diff, or a particular gene family or protein that you want to work on. You could find sequences similar to that, or that code for that gene family, and then go through the steps of downloading the FASTA, looking at the alignment, building a phylogeny, and then talking about it. Because we can't build major phylogenies because we don't have the computational power to do this, I would say you're looking at 5 would be your minimum number of sequences to 10 to 15. You could push it and see how far you can get, and it'll still resolve quickly. But I don't want you to spend forever doing that. If this is ever something you're interested in doing in the future, we can work on that together, and I can give you the computational power you need to really complete those trees, but in the meantime, it's not worth it. Okay, so you're going to look at the kind of lit review you've done of the evolutionary history. You're going to look at this tree and compare how those two things agree or don't agree with each other. And then you're going to talk about what questions you can ask because you know this evolutionary history. So, for instance, I for my PhD, went into a lab that studied cone-headed katydids, okay? They knew that these katydids had different calls. They knew that these katydids could do things like stream segregation, which is where it's how we can see in 3D, or it's also called the cocktail effect. So if you're at a party and you're talking to somebody, but your spouse or significant other says your name, you instantly can hear that too. Or if you're, for the moms in the group, if you are sitting and then your child screams, not only can you differentiate your child from a different child's scream, but you can also instantly hear that in addition to what you're actually discussing. So that's stream segregation. People with schizophrenia can't do that. Katydids that I study can do that. So they knew all these different things. And then I took DNA from each of the different groups, species groups, and populations kind of across Central America and the U.S. And I built so that we could look at the evolutionary history of that group. So then I know how the species are related, and I know where the traits fall. And we'll do this a little bit in comparative methods. And so then we could start asking questions like, why do these two sister species, two closely related species, have completely different calls? And how often do you see that? Things of that nature. So this would be something like when we were looking at the Ebola paper and you saw that the E. coli paper, and you saw that you could get very similar mutations that led to antibiotic resistance, or you could get lots of mutations that were seemingly similar that didn't. Like one group would have it and one group wouldn't. And so the coolest thing I think about incorporating evolutionary history into your study system is you can kind of direct these questions into a better way of testing your overall system and its hypotheses. So you're going to say, what questions can we ask now that we know the evolutionary history? No. What questions do we need to still answer? Like, what's missing from this picture? Depending on what you're choosing, you can talk about the evolutionary mechanisms, like if this group seems to have diversified via drift or selection. That's going to really depend on the system. And then when you're talking about, when it comes to those of you that are in pre-professional programs, I want you to talk a lot about how this changes diagnostic techniques. So knowing that this patient has an infection that is evolutionarily similar to, is infected with a species that is similar to, or a strain that is similar to this other strain, does that change how you treat it or not? So in some cases, I would say absolutely. Like you're going to want to treat a patient that has some certain type of infection with the same way that you knew was effective in this other strain, but not always. And then you also kind of need to talk about how feasible it is to take this evolutionary approach when you're, say, doing dentistry, or if you're pre-vet or something like that. Like, where does this come in and where does it not? Okay. And so a third of your points for this final project are going to be that data processing. So like downloading sequences, building a alignment, doing the phylogeny. So that's not super difficult. But I want you to take it all the way through to like FIML and then visualizing in R. We're going to do more of those steps. And I just want you to have known how to do that. I feel like it's a good skill. I feel like it's a great perspective to take. And then I kind of think it'll change how you view papers and other species interactions in the future. Okay, a third of your points are going to be this lit review that you're going to do on your system. And then a third of your points are going to be those questions. And I'll lay out those questions in print form as well so that you can answer that. But I just wanted to kind of talk you through this system or this assignment. You're going to pick your topic for this assignment relatively soon. Because you're going to pick it so soon and then probably start working on it later, I'm sure your topics will change a little bit as you go. Like, you're going to have to fine-tune your topic. But it'll be interesting to see where it goes. I mean, sometimes you're going to be limited by what sequences of it are available, and sometimes you're going to be limited to what papers are available. But remember that when you're looking at NCBI, the sequences are usually submitted because somebody published a paper, so you can at least find the papers that use those sequences and kind of go from there with your lit review. Okay, I just wanted to give you a brief rundown of what your final project is, and then we'll talk more about it as we go. See you later.